Hi, year 11s and 12s. This is the third video in our first PowerPoint of Stage 2 Biology. So this is the DNA structure PowerPoint. In this last video, we're going to be looking at DNA replication. This will most likely be the shortest of the videos. Again, as with the last video, there is no need to know on the next slide. That's all in the first video. So if you need to write that down, go back to that first video, pause it, write them all down. They're relevant to this video here. Let's get started. Again, if you need to follow up with any questions from this, this all is in the SASTA Workbook Unit 1.1. So DNA replication. Now, there's three different types of replication here. These were all methods that we tested in the past to try and figure out if DNA replication was conservative, semi-conservative, or dispersive. What we came to the conclusion was that DNA is semi-conservative, and we'll talk about that in a second. Don't get too confused with conservative and dispersive. They're just there to help understand why DNA is considered semi-conservative. For now, though, when in the cell is DNA replicated, DNA is replicated right before cell division takes place. That ensures that the daughter cell, so the new cells that arise from cell division, receive an identical copy of the genetic information. So we replicate the DNA to make sure there's two copies for each of the two new cells. DNA replication will occur in the nucleus of eukaryotic cells because that's where we find the DNA, whereas it will happen in the cytoplasm of prokaryotic cells because, as we know, prokaryotic cells don't actually have organelles, so they don't have a nucleus, it just happens in the cytoplasm. The first process is that the DNA strands are separated, and each strand is used as a template for the synthesis of a new strand. Now, when we have that new strand of DNA, it's made up of one old strand of DNA and one new strand of DNA. Okay, so semi-conservative. We split the DNA strands up. One of the old blue strands is used to make the new yellow strand here, while the other blue strand is used to make the new yellow strand here. So it's semi-conservative. One old strand, one new strand in each of the replicated DNA molecules. Okay. Conservative meant that you'd have, once you separated the strands, you made an exact copy, both the old strands stayed together, both the new strands stayed together. That's not how it works. Dispersive, you broke up the DNA into little pieces, and they all merged together with the new and the old, which is a lot of work. Again, not how it works. Our bodies and our DNAs, and all living things' DNA, is the semi-conservative model, where the strands separate, one new strand is made off the old strand, and you end up with two molecules of DNA, one with an old strand, one with a new strand. Again, here's another video. This one, it's called, you can find it up on YouTube, DNA Replication 3D. I'm not going to show it in this video because we'll watch it in class. It'll also be on the useful videos section of the Doing Science Right Year 12 SACE Biology page. What are the steps to DNA replication? Well, the very first step is an enzyme called helicase. Now, just quickly, when we're talking about enzymes, most enzymes end in the word, or the suffix ase, A-S-E. And the first part of the name kind of gives you an idea what it does. So the enzyme helicase, because it sounds like helix, we know that it's actually breaking up the strands of DNA and removing that helix structure, that helical structure of DNA. So helicase is an enzyme that breaks the hydrogen bonds between the complementary base pairs and physically separates the strands of DNA. Once those strands are separated, we get to step 2, where free DNA molecules that are floating around in the nucleus are attracted to those complementary bases and will form hydrogen bonds between the free bases and the template strand. The final step is another enzyme, DNA polymerase. Now, again, ASE. No, it's an enzyme. This time, the first part is DNA polymer, which probably tells us it's going to create a polymer. So in this case, it's joining all of the bases together, and then forming that sugar phosphate backbone. Okay, so replication, very simply, helicase comes along, breaks all the hydrogen bonds, Free DNA molecules in the nucleus are attracted to those complementary base pairs, and then DNA polymerase comes along connecting the sugar and the phosphate, creating the sugar-phosphate backbone, and then the strands wind back up again. And that's it for DNA replication.
That's all, folks.